Well, welcome back. We are looking at Chapter 9 in Bio 121. And what we're looking at, of course, now is how cells actually communicate. And previously, we had introduced this idea that you have different mechanisms that will allow signals to be acquired at the membrane. And we're going to call these passageways now propagation of that signal to be an entire series of steps where we're transducing that information that would requires the presence of those receptors such that some series of mechanisms that will lend to a chain of events that will then cause some integration or understanding of the information so the cell knows how to respond. So the transducing of the signal itself means that somehow we're going to have to change something at the cell membrane so that you can actually have the cytoplasm be where the work is being done. And one mechanism that I introduced to you a moment ago was where you could have receptors that bind to each other that form stable complexes. Now this should be updated because we introduced this idea of two receptors. Well, if it's two sets of receptors, it's still dimerization. And in fact, you actually see that in biology. But the reality is going to be something along these lines. Is they were inactive before they were dimerized. They are active at dimerization. And what's going to happen is in some instances, you're going to send some series of events of molecules that will be activated that will then follow that ligand binding to the receptor. And inside your textbook, they sort of draw these little lines that go like this, response that travels inward, right? Something like that. But the idea, of course, is what that means is they're traveling to specific locations. And if here's a cell here, let's put a receptor here. Even though, say, we actually have dimerized our set of receptors, they're now phosphorylated, what's going to happen is you're going to change a series of events that slowly travel through the cytosol of the cell, but ultimately the nucleus of the cell still needs to know that this is what's happened, and ultimately that will change what's happening in the response of the cell. Okay. Now, how Mother Nature does this is actually kind of cool, right? I think all this stuff is cool, so just, bear with me. So here's an example of the epidermal growth factor receptor family, right? And EGF binds to EGFR. EGF is the signaling molecule. EGFR is the receptor. Okay, and what's going to happen is those phosphorylation systems will now allow a series of proteins, GREB2, SOS, RAS, and GTP, to bind to these entire systems. And what's going to happen is, as a result of the phosphorylation that's occurred on the receptor homodimerization here, you can actually see now a series of cytosolic proteins, MAP kinases in this case, are now actually sending signals inside the cytosol of the cell, where eventually what's going to happen is one of these will now travel into the nucleus of the cell, where it will stimulate cell proliferation, cell migration and adhesion, as well as angiogenesis. And what it's doing is it's going to stop automatic cell death or pre-programmed cell death. And collectively we call that event apoptosis. So notice what's happening here. If this is just one system, EGF, EGFR, there are many other signaling pathways that are similar to this. And what you need to do now is you need to coordinate how it is that Mother Nature will send these signals in, and then where will they go inside of the nucleus, such that it will cause a cell to actually have a specific, now metabolic response. Throw this in here because we know that there are many different types of cancers, right? And one of which can be mediated through the epidermal growth factor family receptor and its family members, ERB2, ERB3, and ERB4. And what happens, of course, is when they are not behaving properly, you'll have uncontrolled cell growth. When we cover this inside Chapter 10, you're going to see that Mother Nature has a specific pattern for how cells will grow. When we see this, when you're looking for images with your next microscopy lab, what you're going to be seeing, of course, is that when things are growing in a regulated manner, it's very tightly coordinated. When you lose that tight coordination, that uncontrolled cell growth is what we actually call cancer. How does this actually happen? And I was thinking about, when was the first time I actually heard about phosphorylation? I was like, I think I had to have been probably, I don't know, sophomore in high school, maybe a junior in high school, thinking to myself, well, how is it Mother Nature can switch these phosphorylation residues? And what she does is she will attach them to serine, threonine, and tyrosine inside of proteins. And each of these represent a slightly different chemical state. And because it has a slightly different chemical state, it means each of the different signaling pathways that may arise from each of these will be slightly different from one another. What's going to happen is we will understand, if we took a cell biology class or something like that, you would learn that there are different kinases that will catalyze the transfer of a phosphate group. And there'll be different phosphatases that will remove that phosphorylation. Okay? So here's the phosphorylation here, phosphorylation there, and this is for serine, threonine, and phosphotyrosine here. So how does this work? Well, in one system, we might actually try something else. Instead of phosphorylating the way we would think signaling molecules would work, sometimes we can take a second messenger system where, let me draw this out for you so it actually makes sense. Okay, here's a cell. Remember, this is a second messenger system, so some sort of signal is going to bind to a receptor. So this is our first messenger. Our second messenger now is going to cause some change inside of the cell, and that second messenger will now signal inside of the cell. That's actually how this works. And an example here is the formation of cyclic AMP. And where did cyclic AMP come from? Cyclic AMP came from adenosine triphosphate. So remember, all that extra ATP we have inside of our cells, remember I told you it could become a signaling molecule. Now you're going to see how. And what it does is it cleaves those phosphates off in the presence of adenylate cyclase. Those are removed. And what's going to happen now is we're going to cyclicize. We're going to create what looks like a circle here. You're like, that does not look anything at all like a circle. And it's not so much that it's a circle, it's now the entire shape and the charge has changed. Now what's going to happen is this will continue and it will signal like this until something like a phosphodiesterase comes along and then unfolds the shape, converting cyclic AMP back into AMP. Okay, so this is the structure of AMP here, adenosine monophosphate. When it's cyclicized, it has one role. When it's deesterized, it goes back to its original shape, and now we can actually add more phosphates to it. Now, why would Mother Nature do this? Well, these series of second messenger systems will create a series of downstream signaling molecules. What's going to happen is, in this example, phospholipase C breaks down PIP, phosphoinositol bisphosphate, into inositol triphosphate and diacylglycerol. Okay? Diacylglycerol, so all of this work, of course, this is all like Nobel laureate level sort of understanding of how things work, right? And what you're looking at is the mechanism that was discovered that actually shows how we break this here, the phosphoinositol bisphosphate, in the presence of an enzyme. So you now end up with two different signaling molecules. And what's going to happen is the IP3 goes off and does one thing, and the diacylglycerol is going to go off and do another. How did we change gene expression as a result now of this slow signaling system? Now, one of the things that your textbook, I think, does a poor job is sort of weaving these stories together. Like, this piece of the story would have been better told, let's see, where was that? Here, right? So remember a moment ago, I showed you how it is the epidermal growth factor receptor causes this pathway to become activated, and here are ERKs that have been phosphorylated, and that ERK now drives cell proliferation, cell migration, and adhesion. And you should be asking yourself, how the hell did that happen? Well, it happened because that ERK now travels into the nucleoplasm. It's traveled inside the nucleus of the cell, and now they give you too much information inside of your textbook. It's like the person who does this, like, this is their pet project, and they just wanted to throw everything at you to see if you can hang on. Well, when we're actually about to translate something, which we haven't gone through yet inside this class, you're going to have to change the activation energy so that we can actually allow protein synthesis to occur. And one thing I taught you is that mRNA structures are supposed to be single-stranded. And I didn't lie to you. But the chemistry of RNA is that, in general, if we want to stop its activity, we create regions of it that are folded. And that folding actually stops molecules from traveling along it. So if we know we transcribe DNA to make mRNA products, that ERK that came in, that was phosphorylated, is now going to add energy to the E1F4E and the MINC1 here, and this will now drive protein synthesis. I'm trying to think of Mark's last name. Mark. He left and went to Brown, right? And what he was doing, he was looking at the mechanism where this actually works. And not that you really care about the mechanism itself, but what you can see here is that a phosphorylation event is now going to allow for this complex to now change its shape, and that causes an unfolding of the mRNA. And because that unfolds, now you can begin to translate what that information is going to be. Why do we do this? Well, it takes energy to build things, and if you don't regulate when you are building things, you waste all of the energy inside of the cell. Okay, so what Mother Nature is going to do is she's going to create a series of systems where what's going to happen is the outcome is going to lead to an increase in the supply of glucose, which would then increase the amount of metabolism inside of that cell. Not addressing where the glucose is coming from. And if this, of course, is too much of a system, okay, where these two signaling, or the parts of these signaling systems could drain all of the glucose inside of your body, another hallmark of cancer. The growth part here, okay, where we actually have a series of hormones that are called growth factors, will bind to those tyrosine kinases, the epidermal growth factor kinases, right? And those epidermal growth factor kinases, okay, will then increase the signaling through that G protein mediated RAS pathway, where the MAP kinases then will change that signaling process. And how does it actually affect the structure? And a really good example can be seen here. I mentioned apoptosis to you as something that was really important, okay? Come on, Jonathan. Apoptosis. Where what's going to happen is Mother Nature will not allow apoptosis to occur here and not allow apoptosis to occur here, okay, but will increase apoptotic events inside these spaces here. So look down at your hands. Look at where your fingers are, okay? And maybe you were born with a little bit of extra folds of skin between that. You call it polydactyly if you actually have extra fingers and stuff. But in the apoptotic events that are actually happening here, what Mother Nature is doing is she's cutting out this tissue. Okay, and by cutting out that tissue, she will allow now a greater range of motion around the joints themselves. Okay, so in this instance, apoptosis is what's going to allow where the cell signals are terminated, okay, by the degradation, and then how they're going to do this, of course, is the phosphatases. I thought I edited that. Okay, phosphatases. And what's going to happen here, of course, now is that you have this interplay between kinases that act in one direction, phosphatases that work in another direction, and you actually have your own feedback loops between what can happen in signaling systems inside of the body. Now that's a lot to sort of wrap your brain around, but let's think about how Mother Nature now uses this inside something really simple. We'll think about now singular cell systems where we're talking about yeast. They can use a signaling molecule called mating factor, right? And this mating factor binds to the cell surface on nearby yeast cells. And what happens is they stop their normal growth and initiate a cell signaling cascade that actually has those G protein systems inside of it. You're like, yeah, what's the big deal? Well, the big deal is this, is there's more than one signaling molecule. There's an alpha and an A. And depending upon what type of signaling molecule was let loose, that will determine how the individual yeast cells activate those G protein coupled receptors and therefore the binding proteins that will signal inside of their cells. That's important, okay? Because now what this does is says, look, if we know we can figure out that there are pathways, how are the pathways different inside of the cells? I'm going to send you away to do a little bit of homework, because inside single-celled structures like bacteria, where in many instances the density of the cells will drive the production not only of receptors, but also the signaling molecules. So when you have a very small number of bacterial cells, you'll have one set of signaling systems happening inside of the cell. And then when you actually go look at populations of high cell density, what happens now is these molecules then change the transcription and translation rates inside of that large number of bacteria. And one way to think about this is what these actually call. So quorum sensing is literally the number of cells that are inside of an area. And once quorum has been identified, it uses a series of molecules that are called autoinducers for this to occur. And what I'd like you to do is I want you to go find anything you can on the formation of biofilms in human disease. And the idea is this. You're going to go discover that bacteria inside your body sort of find different locations. And if you think about what it's doing is it's creating a colonization event. Okay? I know we're thinking about the viral effects of COVID right now, but one of the things that actually happens with bacterial infections is they'll hide inside your body, inside specific areas, until the environment is appropriate for them to actually now do their job. And this is what's actually happening when you're creating things like biofilms. So I want you to just go poke around, see what you can go find. There's some interesting things that happen as a result of the formation of biofilms. And we will return to this idea inside lab. So this completes chapter 9. And if I was to add anything to this, it's sort of now that we've given you these sort of tiny individual pieces of information, sort of take a step back and look at the entire story and ask yourself, oh, do I understand that even though these are all mechanisms and they might be being used at any single given time, what Mother Nature has to do is she has to figure out a way to differentiate the signaling systems that are moving into the cell. Okay? And this is a very important idea because depending upon the type of signaling molecule that binds a unique receptor at some unique location in a cell, you're going to change then how that cell will respond. So let's get back to the original example I gave you. When you're trying to have a conversation with somebody, think about what it means to use nouns, verbs, adjectives, adverbs. Now, I'm not saying that these are analogous, but hopefully that helps you understand that beyond saying hello world, you can then use different materials to create different ways to communicate between cells. And they will still use those 4 basic characteristics we spoke about at the beginning of chapter 9. And you can see those here. You can have many different molecules that use autocrine signaling. You can use many different molecules that will use endocrine signaling. So sort of build some complex understanding, even though we haven't told you how to do this, sort of go back over this idea and think that Mother Nature has figured out how to create these really streamlined mechanisms. And then by adapting the differences, the type of molecule or the receptor or where the receptor is located, that cell will now change its responsibility. And that's where I'll leave you.